So hello everyone, I'm Jasmine Trigg. I'm an associate scientist at Sartorius, and today I'll be discussing quantifying morphological change using label-free live cell analysis. So after a quick introduction into live cell analysis and kinetic monitoring, I will cover our new IncuCyte Advanced Label-Free Classification software module. And I'll also present some neurodegenerative biological paradigms that it can be applied to, where we see morphological changes in our cells, for example, in cell health or differentiation assays. So firstly, what is live cell analysis? In simplistic terms, it is continuous cellular measurement. So using specialized live cell analysis systems, we can monitor our cells throughout all research phases, from their culture to their manipulation, and of course, all throughout an assay time course. So this provides relevant information in real time and enables data acquisition from living cells in a non-perturbing manner. And we can use this to answer multiple research questions. So data I'll be showing you today was acquired using the IncuCyte live cell analysis system, which is shown here. And this allows us to continuously acquire images of our cells from within an incubator in physiologically relevant conditions. And because of this, it is ideal for the long-term morphological analysis of our cells. So traditionally, live cell analysis has provided biological insight using simple kinetic readouts. So these can be based on phase measurements, such as label-free monitoring using Confluence, or on fluorescence measurements. So in the example shown here, where we can quantify phagocytosis in microglia through an increase in a fluorescence signal of a pH-sensitive dye. So over time, these readouts have moved from being population-based to focusing more on individual cells on a cell-by-cell basis. And this has really allowed us to interrogate things further. For example, using cell-by-cell analysis, we can gain morphological information on cells using univariate morphology readouts, such as cell eccentricity or cell area. Although these are insightful, they act as a surrogate for total cell morphology, as they can be limited when we're trying to quantify complex and diverse morphological features of cells. However, the development of advanced machine learning algorithms has facilitated the development of more complex quantitative label-free methods that, in combination with live cell imaging, allow us to monitor kinetic changes in cell morphology. For example, we can perform counts of viable versus apoptotic morphologies, or this allows us to monitor dynamic changes that occur during differentiation, as exemplified in the video here of iPSC-derived microglia. This advanced analytical approach uses multivariate data analysis, and this is an unbiased and very thorough way of quantifying cell morphology, as instead of using a single metric, it incorporates a number of different features of cells, such as the shape, the size, or the texture. And some of the metrics that we can use for this are shown here. And what this really allows us to do is adapt this to a wide range of different cell morphologies. So this does sound quite complex, and therefore we have developed the IncuCyte Advanced Label-Free Classification software. And this allows us to quantify cell morphology using kind of more simplified methods. So with this, it enables you two user-defined classes of cells with distinct morphologies to be identified and quantified over time. So in the next couple of slides, I'll go through the workflow of this software and also present some of its applications. So we've built the advanced label-free classification of cell morphology into a simplified and user-friendly workflow. So the schematic shown here is an example using the label-free live dead assay. So first we can acquire our images using the IncuCyte, and then we can segment the individual cells using our cell-by-cell analysis. With the advanced label-free classification add-on, this allows us to process the images using our advanced algorithms and quantify the different features of the cells. And And actually, as part of this, we incorporate over 20 different metrics. Using a classifier, we can then train using two control populations. So in this example, we'd have images of alive control populations and dead control images as well. So once trained, this classifier can then be applied to all of your wells across the time course, and then we can get population data from this as well. So I'll go into a bit more detail into the classification workflow on the next slide. So as I've mentioned before, after image acquisition and cell-by-cell segmentation, the next step is to train the classifier. And this is really the most critical step of the workflow. So shown here, we have our two classes of interest. We have live cells and dead cells. And we can use images of these control populations to train our classifier. And all we really need to do for this is have good representative images of both classes. And this also means that in this previous step, we need to segment them really well as well. So in order to train the classifier, we have two workflow options. So the first is our default workflow, and this uses the whole image. And we've got some smart default settings within this to make it really easy to do. So this is based on homogeneous populations. For example, in our dead control images, we'd need at least 90% cell death for this to work. If this isn't the case, for example, if you've only got 50% differentiation within your well, we need to be able to select certain cells within the image to train our classifier. And for this, we can use the advanced training set selection or refinement mode. And although this is a little bit more time consuming, it has the advantage of giving you ultimate control over your training data set. So what multivariate analysis does here is it allows you to distill a number of different morphology metrics onto a single axis. And this gives us a meaningful score value on a range between 0 and 1. So in this example here, the live cells would have a score closer to 0, and the dead cells would have a score closer to 1. So when we apply this to other images, as you can see here, every individual cell is quantified based on its morphology and assigned a score value on this range. We can then apply a threshold, and this then classes the cells into either one of these classes. For example, with a threshold of 0.5, any cell that has a score below that would be considered live, and any cell with a score above that would be considered dead. And then we can run this across the time course and other wells and get time course and concentration response data from this. So in the example shown here, we can see the percentage of dead cells over time in response to a concentration range of camptothecin. So next, I will show some validation data that we've generated in-house. So here we use the label-free live dead assay to quantify cell health in a neurodegenerative model. The SHSY5Y neuronal cell line was treated with a concentration range of okadaic acid, and this is a dual protein phosphatase inhibitor that is commonly used to study Alzheimer's disease. These cells were treated in the presence of the cell health reagent Annexin-5 to act as a comparison. We then quantified the number of dead cells using both advanced label-free classification and a classification of the Annexin fluorescence. For the advanced label-free classification, we used vehicle wells across all time points to train for live cells, and then we used camptothecin, which is our positive control, at the final time point to train for our dead cells. And there's some representative images of these control populations shown here. So if we look at the microplate view from both the classifications, we can see that there was a similar kinetic profile across the plate for both methods. Transformation of this data into concentration response curves showed that both methods, that there was a cytotoxic concentration-dependent effect of okadaic acid on the neuronal cell line, with similar IC50 values being observed. The data also shows that even for difficult-to-mask cell types, such as the SHSY5Y cell line, the advanced label-free classification is able to provide a label-free readout of cell health without the need for a fluorescent cell health reagent. And this means that it's highly suitable for any sensitive cell types that you have. So the activation and differentiation of immune cells is often accompanied by morphological changes. So here we demonstrate how, using the label-free differentiation assay, we can distinguish these different phenotypes and quantify differentiation over time. So here, primary monocytes were isolated and they were differentiated to macrophages. We then activated them to either an M1 or M2 phenotype using cytokines. So as you can see, the monocytes are small, round, and semi-adherent. And then upon differentiation activation to an M1 phenotype, we have this change in morphology with an increased adherence. And it's quite a homogeneous population. We employed the advanced label-free classification. And as you can see, the resulting segmentation here, we have those classified as monocytes with the pink outline and the M1 macrophages shown with a blue segmentation outline. We can then use the results in this classification to quantify differentiation over time, which is shown here as the percentage of macrophages. And interestingly, in this case, we saw that the differentiation process was non-linear. For the M2 macrophages, we saw that there was a more heterogeneous mixture of morphologies. So as you can see in the image, we had more ramified cells, which were longer and thinner, and then the amoeboid, which were rounder and flatter. And so we used the advanced training set selection to manually select these cells of interest and use this to train our classifier. And again, with the resulting segmentation, you can see that we're able to distinguish these two different phenotypes. We've got the amoeboid segmented in pink and the ramified shown in blue. And of course, we can quantify this as well. So the fact that we're able to distinguish different phenotypes, as shown here, is really interesting and quite exciting. I hope you can appreciate how, when thinking back to the videos on the first couple of slides, this has the potential to add even greater biological insight into neural cell types. For example, when assessing the differentiation of healthy versus perturbed microglia, or if we combine this with more functional readouts, has the potential to show if a specific microglia phenotype has a greater capacity of phagocytosis. So just to wrap up, so through our advanced label-free classification module, we can conduct unbiased label-free analysis of morphology. And we can do this using our purpose-built software, using machine learning algorithms. And this allows us to obtain robust and reproducible data. So I've exemplified the use of the advanced label-free analysis for the assessment of cell health and differentiation, although it's worth highlighting again here that this analysis is really versatile and can be applied to anything as long as you have two classes of cells that have a distinct morphology and can be well segmented. And some further applications of this analysis is shown in the table here. So overall, live cell analysis and intuitive label-free analysis have enabled non-perturbing morphological quantification of neural cell types. And this has the potential to deliver greater insight into neurodegenerative disorders. So if you'd like any further information, we have lots of material available on the website. We also have a booth today, so feel free to visit us. And then myself and two of my colleagues will be presenting a poster later on this afternoon. So that was it from me. Thank you for listening.